Okay, it seems a little sparse in here today. I don't know if it's the rain or what. But remember, the advantage to coming to class is that you can ask questions. Okay, this is probably, in my mind, the most conceptually difficult lecture that we'll have all semester. So I'm assuming that we're going to get to the close to the end of chapter 16 here. So don't be afraid to stop to say, even if it's just, can you say that again? Or, you know, don't be afraid to raise your hands and ask questions as we move forward here. So, okay. So last time we started talking about kind of the parts of what we would consider where these adaptive immune reactions are taking place, primary lymphoid organs, secondary lymphoid organs, you know, what happens in a lymph node, what is lymph. We started kind of defining what an antigen is and what kind of molecules make good antigens. In other words, what kind of molecules elicit a really strong immune response. So that brings us to talking more specifically about B cells. And of course, we'll get to what B cells actually do. And then we'll talk a little bit about T cells and what they do, and then kind of finish up with how they both get activated to do what they do. All right, so B cells, where are we going to find these? Remember, these are lymphocytes. We're going to find them primarily in the secondary lymphoid tissue. So that's primarily places like lymph nodes, spleen, and they're really cycling back and forth between that secondary lymphoid tissue into the bloodstream, back into the lymph. Okay, they're cycling all around. In this picture, so right, the B cell is down here, right, and this graphic is showing the surface of the B cell. And a very important part of that B cell is that B cell receptor. There are, keep in mind, you know, picture like this, it shows you one B cell receptor, but keep in mind there are thousands, up to maybe 500,000 B cell receptors on the surface of one B cell. Okay. So, this picture doesn't really do it justice. Picture B cell receptors coating the entire B cell. The parts of that B cell receptor. So there's kind of a transmembrane part. There are two what we call heavy chains. That's just because they have a higher molecular weight. And then there's two light chains, lighter molecular weight. And the business end here is this variable region at the end of both, whatever, the Y against the ends there, that's the part that binds to antigen. Okay, so here we have, pretend this is maybe like some kind of bacterial cell. Remember, there's going to be all sorts of different proteins on the surface of a bacterial cell that would make really good antigens. And then an epitope is a part of an antigen. Okay, so something that's not self that the B cell receptor is going to recognize directly and bind to that variable region of the B cell receptor. Each one of these variable regions has the same specificity. Okay, let's say all those B cell receptors have the same specificity. Remember, we said that's a real hallmark of adaptive immunity. Each B cell receptor on the entire cell recognizes the exact same molecule or motif, we could say. And it's kind of like a puzzle piece. You could think of it. This is 3D and very specific epitopes of antigens. And it seems like, to me, I mean, after I thought about this a while, it seems like, wouldn't it make more sense? Our body, each we have maybe just a few B cells. We have a lot of B cells, but we only have a few. We have a certain kind of each receptor. So we have a B cell receptor that is going to recognize just about any antigen we could ever come in contact with. So we only have a few cells that has each receptor. And it seems like to me, well, it takes a long time for those cells to match up with that antigen if we ever get exposed to it. So why wouldn't we have a B cell that has all different B cell receptors, and then it would very easily come in contact with that antigen? But it turns out that this method is best because when that B cell receptor comes in contact with that epitope, it needs to send a signal down to the B cell in order to act, okay? And just one B cell receptor recognizing one epitope is a very weak signal. You need to get signaling from lots of B cell receptors all around the cell to get it kind of primed to act. So that's why all of those B cell receptors have the same specificity. Okay. So a little bit more about B cells in general. We'll mention here very briefly how they are activated, but we're going to come back to this later. They can either be acted by the antigen themselves and not needing any other stimulation. So we say that it is independent of T helper cells. Okay, the much more common way is that B cells are dependent on stimulation by T helper cells. Okay, and it depends what kind of antigen is being recognized by the B cell receptors as to whether that antigen can activate the B cell right away or whether the B cell needs additional signals from a T helper cell. Okay, so we're going to come back to that after we talk about T cells. So stay tuned there. But what happens once a B cell gets activated? Several things. It's going to get bigger and it's going to start dividing rapidly, and it's going to differentiate into antibody secreting plasma cells. Okay, into the type of cell that's kicking out lots and lots of antibody, as well as some memory cells. So the advantage there, those memory cells are hopefully going to persist for long periods of time. And the next time that B memory cell comes in contact with that same antigen, it doesn't need to go through a complicated activation process. It's already ready to go, ready to start kicking out antibodies again. Okay, so here is a picture of a plasma cell from your book. I think this comes from a guinea pig. I love this picture because, you know, back maybe in 9th grade when you learned first about a eukaryotic cell, you started learning about the organelles, and you had a cell with a nucleus and a lysosome and a Golgi, maybe one of each. This picture makes us realize, okay, that's not really how most cells look. It depends what kind of cell it is. Okay, so here's a B cell that's differentiated into a plasma cell. You can see a lot of, you know, it's a pretty big nucleus that takes up a big chunk of the cell. And then most of the rest of the cell is taken up by rough endoplasmic reticulum. So remember rough ER, it's rough because it has ribosomes studded into the surface. So what happens at the ribosome? That's where proteins are made. Not just any proteins in the rough ER, that's where proteins are made that are destined to be secreted outside of the cell. Okay, so they pass through that rough ER into a Golgi that, you know, usually puts them in some kind of endosome and shuttles them outside of the cell. So full of ER, a few Golgi. I think these other blue blobs are mitochondria because the cell needs energy to make this all happen. But a plasma cell, its job is to be kicking out proteins, and in this case, right, antibodies. Very high metabolic activity, kicking out about 2,000 antibodies per second. All right, so this is a antibody factory. They don't typically live very long because they have such a high metabolic rate. Okay, so they kind of burn out after a while. We think there are different kinds of plasma cells, ones that maybe persist for longer. Okay, and that name plasma cell, I think sometimes that's a little confusing because we've thought of, oh, plasma, that's the liquid part of your blood. Well, I think they called these plasma cells because when they first looked at these under the microscope, they could see the nucleus and then there was lots of plasma. So they called them plasma cells. When you look at a B lymphocyte, and I showed one earlier, a B lymphocyte, the nucleus takes up like most of the cell like that. So it looked like plasma cells had more cytoplasm, and that's why they call them plasma cells. So nothing really to do with blood plasma there. Okay. So let's talk about these antibodies that the plasma cells are kicking out. Let's look at their structure, the different kinds of antibodies that we see, and then what they actually do. There's about 5 different ways that they actually act to hopefully resolve infections in our bodies. So another term that might be confusing that you've heard is the term immunoglobulin. Okay, apparently those two terms are the same thing. An immunoglobulin is the same thing as an antibody. As far as I can tell, I've looked that up several different sources. Some places say, oh, an antibody is a kind of immunoglobulin, yet all immunoglobulins are antibodies. So same thing. I usually use the term antibodies. And if you look at the structure of an antibody, that should look familiar because it looks exactly the same as a B cell receptor. So the antibodies that a B cell makes, it's going to have the exact specificity of the B cell receptor that recognize the antigen. Okay, so we've got 2 heavy chains, we've got 2 light chains, just like a B cell receptor. This fragment, this part at the top, sometimes we refer to that as the arm. So if you sticking your arms up like this, it's also referred to as this FAB. Okay, in other words, fragment of antibody binding. Okay, fragment binding. Okay, there's a hinge region where apparently the protein is a little bit flexible, kind of like you are at your waist. And then the stem region, that's also referred to as the FC portion. That stands for fragment that's crystallizable, in case you're wondering. So the FAB and the FC is the stem. Of course, the part where antibody binds, that's incredibly variable. We'll talk about that variability in a minute. And then the stem region, there's only 5 different stems. So when you look at the kind of the loci on the gene that is responsible for encoding antibodies, there's only 5 different possibilities for the stem, and that antibody is going to be whatever part of the gene that gets transcribed and translated. So if it comes from the mu region of the gene, when you're talking about a segment of gene, it's correct to italicize it. That's why it's italicized there. Then that turns into a IgM antibody. Okay, with that mu stem. If it's from the gamma region, then it's an IgG. If it's alpha, it's IgA. So immunoglobulin M and immunoglobulin G, so on and so forth. So we'll talk about some of these different isotypes of antibody are good at certain things, and other isotypes are better at other things. I like to show this space filling model here. a space filling model, that just means that each atom of the molecule is represented as a little circle there. And that just kind of helps illustrate a little bit the incredible specificity of those antigen binding sites. 3D, like a puzzle piece, it's only gonna recognize one very specific epitope. Okay. All right, so what do they actually do? Okay, 5 different functions. One major function that we just sum up and call it neutralization. Okay, so that just means that the antibodies are going to block adhesion molecules. On either the cell surface or the virus. Okay, so when we talked about, the process of infection, whether you have a virus or a cell, there's always going to be some adhesins on the surface that help it to target what kind of cell is going to get invaded, right? So if you've got antibody coating the outside of that bacterium, say, and this isn't really drawn to scale, the antibodies are way smaller than that. But you can block those adhesion proteins, so the protein can't attach, the bacteria can't attach where it needs to. If we have antibodies to those spike proteins for the virus, antibodies to COVID-19, then it can't bind to those receptors deep in the lungs that it is going to want to attach to. Same thing with a toxin. You can totally, make a toxin not able to function. You can coat it with antibodies. This function we've actually talked about already, activation of complement and inflammation. Okay, so it's kind of a small diagram there. Maybe I can make that a little bit bigger. So here we've got the bacterial cell, the antigen, which is drawn kind of weirdly there in yellow, and then an antibody here is bound to that antigen. That antigen antibody combination actually acts to activate that C1 complex. Remember that we call that the classical activation pathway of complement. Okay, so it's actually something in the stem of the antibody, the Fc region of the antibody activates that first protein, C1, and in the classical pathway, activation pathway. Of course, then we get things like increased inflammation, increased chemotaxis of cells, opsonization by complement, as well as membrane attack complexes. So opsonization, hopefully you recognize that term. That just means that antibodies can bring about increased phagocytosis. Okay, so when we talked about the steps of phagocytosis last week, we said that certain things can act as opsonins, meaning make phagocytosis more likely to happen. And those two things were complement protein, especially that C3b bit that binds to the pathogen, as well as antibody. So you can see here, if this is the pseudopod of a phagocyte, like a dendritic cell or macrophage, there are stem receptor proteins on the outside of that phagocyte. Okay, so it's going to be able to hold on to that Fc region of the antibody. The antibodies are like handles for that pseudopod to grab a hold of. Going to make it more successful in phagocytizing that pathogen. Oh, question. Yes, repeat that part about the handles. So there are Fc receptor proteins in all around the outside of the macrophage. So remember the FC, that's the stem region of the antibody. Okay, so the macrophage, for example, will grab a hold of that stem portion of the antibody, which is attached to the pathogen, okay, and make that pathogen more likely to get phagocytized. Okay, so in other words, the phagocyte can attach to the pathogen much more easily if the pathogen is coated in antibody. And two more ways that antibody works, agglutination. So if something is agglutinated, it means it's clumped up. Okay, and we've mentioned already that our lots of aspects of our immunity, they tend to function better. They tend to notice larger molecules better. All right, so if you have lots of the pathogen that's all clumped together because you know, one binding site is on one cell and one binding site of the antibody is on another cell, you can get these great big clumps of pathogen, whatever, virus, toxin, whatever, and it's going to be more easily noticed, more easily filtered out, either in the lymph nodes or the spleen. Okay, so that gets to why it's important that both antigen binding sites have the same specificity, because you want them to all notice the same thing and make a big clump. Okay, and the last way an antibody can actually a little bit more directly bring about the death of a pathogen in conjunction with a NK lymphocyte, a natural killer lymphocyte. All right, so when an antibody is bound to a pathogen, a natural killer cell can again recognize that it will have receptors for that stem portion of the antibody, and that can trigger the natural killer cell to release cytokines that act directly to kill the pathogen. Okay, so the NK cell releases cytokines. Okay, so those two cytokines, perforin and granzyme, that's the same mechanism that cytotoxic T cells sometimes use to kill virally infected cells. So we're going to come back to that and talk more about that later, but it's the same mechanism again that natural killer cells can use when they've been stimulated to do that by recognizing an antibody, right? And we call that antibody-dependent cellular cytotoxicity, so ADCC. Lots and lots of abbreviations, TLRs, PCRs, BCRs. You gotta, to some extent, to be fluid in this field to just recognize all those different abbreviations. All right, so let's talk about the different kinds of antibodies, and we'll at least mention to some extent, once you look at the structure of some of these different 5 different isotypes, you kind of understand how some kinds of antibody are better at some of those functions than others. Okay, so the first one we'll talk about are the IgM antibodies, because they are the first ones that are produced when that B cell gets activated and changed into a plasma cell. It always starts pumping out IgM antibodies first. Okay, so it's the first isotype produced. Okay, and interestingly enough, usually 5 of them are bound together to make what we would say a pentamer. Okay, usually that's the way, that's the form that takes. So 10 different binding sites when you put it all together like that. So really good at agglutination, because there's 10 different binding sites. It can bind to 10 different cells, potentially. Usually not that many because it's pretty small molecule still, but all right. And it's good at some of those other functions as well. It's really only a fairly small percentage of the total antibody in the blood. And one other thing to mention, how do those 5 antibodies come together? It has something to do with this accessory molecule that we call a J chain. This is just a little protein. That's what this little, right in the center there, that little arc, that's supposed to be the J chain. We know that that has some role in binding those 5 IgMs together. Not quite sure of the mechanism there, but okay, we don't find that when antibodies are not bound to other antibodies. So, okay, so I said that there's actually not a lot of IgM in the bloodstream. The reason is that once you get to a certain kind of point in the infection, those B cells typically get cytokine signals to switch to a kind of antibody that may be better suited to fight that particular infection. Okay, so again, this is due to cytokine signals. Oftentimes we get some class switching. So it starts out with IgM, and then after a while, those plasma cells start making a different kind of antibody, but the specificity of the antibody remains the same. Okay, so it's only that stem region, that FC region, that changes. And most often, it's going to change to an IgG stem from that gamma part of the gene, because most of the antibody in circulation in the blood is going to be IgG. So that's kind of the most famous thing that you would want to know about IgG, that it's the most common kind of antibody that we have, partly because it's the longest acting. It seems to stay circulating for longer than other kinds of antibody. And the other famous thing to remember is that it's the only antibody to cross the placenta. So I don't know, I was trying to think about ways to remember that. IgG, goo goo or something, baby, I don't know, might be a way to remember that. But so it's really important for the developing fetus to get some antibodies from the mother, because in its first few months of life after birth, it has a very immature immune system. And so it's going to, those antibodies that the mother has been, to things that the mother's been exposed to, is going to help protect that fetus in the first few months of its life. The one drawback to that is in the case of Rh disease. So we could talk about this a long time because it's kind of interesting, but of course, some of us have that rhesus antigen and some of us don't, right? So if you have A positive blood, then positive means you have the rhesus antigen. If you have A negative blood, then you're negative for the rhesus antigen. So the baby is not necessarily have that same plus or minus as the mother, right? It might have what the father has. So Rh disease can develop if the mother does not have that antigen, and usually during the birthing process, maybe the mother and fetal blood might mix a little bit. So after the first pregnancy, a mother can be sensitized to that antigen and start making antibody to that rhesus antigen. So it's usually not a problem for the first pregnancy, but on subsequent pregnancies, if the next baby is negative for that antigen, positive for the antigen, and the mother is making antibodies to it, then that can cause hemolytic disease in that newborn. It can cause death, sometimes just a mild anemia, but sometimes it can kill the baby. So luckily, we have something called RhoGAM shots, which are administered to women at some point during the pregnancy and right after birth of babies to help those mothers' antibodies be neutralized. Good prenatal care is always a good thing. Okay, so these IgG antibodies, the structure there kind of looks very similar to B cell receptor. They function in all those different functions of antibodies. Okay. IgA antibody, what's kind of special about IgA? Two things. It often exists as a dimer form, so two of them together. And it is the antibody that's most plentiful on our mucous membranes. As well as bodily secretions. So there's only like maybe 12% in the blood, okay? But it's the most prominent antibody on places that are really common portals of entry for pathogens, like our respiratory tract, that's a mucous membrane. Our gastrointestinal tract, that's a mucous membrane. Okay, so there's this secretory component, all protein that apparently helps that antibody be transferred to our mucous membrane. There's also a J chain that just helps those two antibodies bind together. Okay, so what kind of bodily secretions? I didn't define that very well. Things like tears, breast milk. Saliva. Okay, so this is the kind of antibody that new mothers that are breastfeeding will pass on to their baby. Okay, so IgG passes through placenta, IgA through breast milk. Okay. And again, pretty good at agglutination because there's 4 different binding sites here. A factor that can lead to neutralization as well, the pathogens. And then I put IgE and IgD on the same slide because they're not really very common and it's not really understood very well what they do. IgE, less than 1% of the total antibody in the blood. So, like I said, we don't have very much of it. We do know that it has some role in fighting parasitic worms. So same thing with, remember that granulocyte eosinophils has a big, I think, a prominent role in fighting those parasitic worms. So it kind of works together with eosinophils to fight those kinds of pathogens. And we do know that IgE is much more common in allergic reactions. So it appears to maybe cause a little bit more immune response than what's really appropriate sometimes. And IgD, also very uncommon, and we think it actually functions as B cell receptors. So we don't really understand a lot more about it, but it apparently is just an antibody that never was secreted from the B cell. So it stays on the surface of the B cell. Like I said, we don't really understand their function very well. Some mammals don't make IgD antibodies. So again, that's pretty much all I have to say about that. And there are other kinds of isotypes in other mammals. Sharks have IgW, for example. So there's lots of other stuff out there. Okay. Oh. All right. So one issue that I just want to kind of give you the basic idea of is that I've alluded to this. Like we have only a few B cells with each B cell receptor. So we can recognize, we already have B cell receptors to recognize just about anything that we would ever come in contact with. How is that possible? So we only have about 25,000 genes. So if each B cell receptor, was made from one gene, we don't have enough DNA in our body to make that many different variations of B cell receptors. So This is a question that plagued researchers for a really long time. We finally figured out, or some Japanese scientists figured out in the 1970s, that amazing diversity is generated by recombination events. So the idea of this, which is from your book, you can read more about it if you're interested. But the idea is that the parts on the gene or on the chromosome that encodes for B cell receptors, basically, it's kind of like a deck of cards. So you get like 3 different cards out, and that makes your B cell receptor. Okay, shuffle the deck. Okay, get 3 other cards. There's your B cell receptor. So basically, these are recombination events. An enzyme goes down and picks one of these V regions, and one of these transcribes one of the parts of the D region, and one of the 6 different J regions, puts them together and makes a B cell receptor. So there's many different combinations that are possible that makes up that variable region of the B cell receptor. So let me just write that in. Recombination events generate diversity, generate B cell receptor and T cell receptor. We haven't talked about those yet, but it's kind of the same mechanism. So with these different recombination events, and you've got that happening on the heavy chain as well as the light chain, and you've got 2 different alleles. So like doing the math and kind of, taking all that into account, they estimate that we have maybe 10 to the 23rd different possible B cell receptors. So that's a lot, okay? So again, the idea is that we have those for anything that we would come in contact with, and then we make more of them if we need them, okay? Not that we fashion a B cell receptor to fit whatever we come in contact with. We don't think it works that way. Okay. All right. Oh, so good question. RAG, that's the name of the enzyme. It's a RAG protein that goes along and picks out the variation of each one. So yeah, good question. All right, so let's move on to T cells. We'll circle back to B cell activation. All right, so remember T cells, they're primarily made in the bone marrow, but then they go off to the thymus to mature into different kinds of T cells and to get their T cell receptors. So there's 3 different major kinds of T cells, cytotoxic T cells. Okay, what they do when they're activated is kill our own cells that are infected with some kind of intracellular pathogen, like a virus. Okay, so these are, if antibodies are cheap bullets, cytotoxic T cells, they're like grenades. Like they're doing a lot of damage because they're, again, targeting our own cells. Okay, they have a glycoprotein receptor called CD8. So CD8 positive. So that stands for cluster of differentiation. It's just they have a T cell receptor, but then they have these other receptors that we call CD8. Okay, and it just, they have some function, but also it helps us recognize, oh yeah, this is a cytotoxic T cell and not a helper T cell. Because helper T cells have a little different cluster of differentiation. They are CD4 positive. Okay, so maybe you've heard of CD4 positive T cells because that's the population of cells that HIV targets and decimates in an AIDS infection. Okay, so helper T cells are famous for kind of coordinating lots of different aspects of our immune response. Okay, you can think about them as kind of like the general in charge of the troops. Okay, so when those cells are damaged, that really has devastating effects on our whole immune response. There are actually two different kinds of T helper cells, and we'll see like when they differentiate. The T helper ones drive the activation of cytotoxic T cells, and we'll get to that in a minute in a graphic or in a few minutes. And T helper 2 cells are going to be important to activate B cells to make antibodies. And there's a third kind of T cell that we're not really going to talk about anymore. I'm just going to mention them here, but there are these T regulatory cells that act apparently to dampen the immune response when that's necessary. And the mechanism for that is, I don't really understand it, so we're not going to talk about that anymore. But there are some mechanisms just, you know, to, again, dampen the response when it gets to be too much. All right, so looking at a T cell receptor, I thought it might be helpful to compare a T cell receptor to a B cell receptor. There's some similarities. They both have a transmembrane portion, no heavy and light chains for the T cell receptor, but there is here an antigen binding site at the top. I don't even know why these carbohydrates are labeled. I don't even know what function that they have. But there's a variable region and a constant region. One major difference between a T cell receptor and a B cell receptor. A B cell receptor, remember, recognizes antigen directly, or epitopes of antigens. Okay, you can see that right here. It's binding right to that epitope. T cell receptors can't do that. Okay, T cell receptors, they're a little bit more particular. So they only recognize antigen when it's presented a particular way. When presented by Mhc glycoproteins. Okay, so they're very particular. They cannot bind directly to an antigen. They have to have it presented to them in a special way. Okay, so let's look at that special way that that's accomplished. Okay, these Mhc glycoproteins, I'm gonna come back to this and just make sure I talked about all those things, but I think it's easier to introduce when we have a picture in front of us. So I'll come back to that in a minute. So we have genes, we have Mhc genes that make the Mhc proteins, glycoproteins from the genes. And all of our cells in our body that have a nucleus have Mhc class 1. Okay, so there's two major kinds of these Mhc glycoproteins. Okay, every cell that has a nucleus will present antigen with Mhc class 1. Okay, so think about the cells that don't have a nucleus. The only one that I know of, there might be other ones, are red blood cells, right? They don't present antigen with Mhc class 1, but every other cell has this mechanism. And this is a mechanism that cells have to advertise on their surface what's going on inside their cell. Okay, so this is happening normally all the time, and we're going to look at kind of the basic mechanism here. But little bits of proteins inside the cell are being sampled and presented on the outside of the cell. Okay, normally that's ignored by our immune system. But if that cell is infected by a virus, then it's going to pick up bits of that virus and advertise that on the surface. Okay, so let's say advertises what's going on inside of the cell. Okay, so it's the mechanism that our, in this case, cytotoxic T cells have to know if they need to kill that cell because it's infected by a virus. Okay, otherwise the virus is inside the cell and it might not leave any evidence on the outside that the cell's been infected. Okay, so I think I have this written somewhere else, but it actually presents to cytotoxic T cells. Okay, MHC class 2 then are only going to be used by antigen presenting cells. So that's, remember, macrophages, dendritic cells, and actually B cells. And we'll talk about a little bit later why that is, because we haven't talked about B cells as being phagocytic, and they're not really exactly. Okay, so these antigen presenting cells, they have MHC class 1. They also have MHC class 2. So the MHC class 2 are a way for them to advertise on the surface of their cell what they've just ingested. Okay, so they phagocytize the pathogen, they chewed it up, and then those bits that they chew up, they show on the surface of their cell. Okay, so this is the way that they go looking for lymphocytes to, get involved to make some specific response to help fight the infection. Okay, so they are actually presenting to T helper cells. Okay, so that's going to be things that they phagocytize. Those are termed exogenous antigens. They were outside of the cell. And things that were inside of the cell, endogenous antigens. Okay, so I think I have that written back up here. The difference between the two classes, present antigen to cytotoxic T cells, again, present antigen to T helper cells. Okay, what else did I say here? They present antigen to T cells. So this seems like it's a little bit out of left field. So variation in MHC in the antigen they present play a role in transplant rejection. Okay, so those MHC genes are what are called polymorphic, meaning we all have slightly different variations of that gene. Somebody in your family, you're more likely to have very similar MHC genes, but to people on the street, they're often very different. So essentially that means that those MHC glycoproteins will present slightly different antigen. The receptor, they're recognizing, different bits of amino acids. These are just like 12 to 15, maybe a series of 12 to 15 amino acids that they're presenting on the surface. And the ones that I present, these antigens that I present, may be very different than the ones that your MHC recognizes and presents. So two people, they're both healthy, they're the same age, they're exposed to the same pathogen, the same dose. One person might get very sick and the other person may be very mildly sick or not sick at all, just because differences in how their body kind of processes and presents that antigen. Okay, and that's one of the things that we look for when we're looking at organ transplant. Is we try to match that up as best we can between the donor and the recipient. It's more likely to be a 6 vessel, less chance of rejection. Okay, so a little bit about kind of the mechanism of how this works. Maybe it might help you picture this, but these are the graphics from your book. So with that MHC1, again, sort of the basic gist here is that a random sampling of proteins within each of our cells gets catabolized, gets broken down. And you would think that would be bad for our cell, right? But apparently it's just a little bit here and there, and our cell doesn't really notice. There's a little kind of what we call a proteasome complex that does that. So puts little samples of our proteins, enters into the ER, endoplasmic reticulum, where our MHC proteins get expressed. And then, you know, there's different ones that different MHCs are going to match up with different sequences of amino acids, but they pinch off from the ER, travel through the Golgi, through an endosome, and then gets presented on the outside of the cell for a time. If they're just self-proteins, remember, they get ignored by our immune system and eventually they degrade. Okay, so MHC1, one of the ways that I like to remember this is, you know, one, when you raise your hand, right, you're volunteering what's going on. So MHC1, the cell is just volunteering, hey, this is what's going on inside my cell, just in case you want to know. Okay, with the MHC class 2, remember, this is only on our dendritic cells, macrophages, and B cells. They are phagocytizing some pathogen. The MHC class 2s pinch off from the ER and then fuse with the phagolysosome. So after that pathogen has been consumed and, you know, chopped up, different epitopes will match up with the MHC, and then again, it gets expressed on the surface of the cell. Okay, so with the MHC class 2, with two hands, you're handing something to me, like, here, this is what I'm dealing with. Okay, these antigen processing cells are now looking for a T helper cell with the same receptor that they're willing to help start up a more specific reaction that will work against that particular antigen. Okay, and there's also a pretty good video with that as part of your homework. So pay attention to that, and that will hopefully reinforce what we're talking about. Okay, so just to check, see if you're paying attention here, a little quick question. All nucleated cells in the body have the ability to advertise endogenous antigen on the surface of their cells by means of, I actually put this on a test once. Nobody said Facebook. So that's by means of the MHC class 1. Okay. All right. Okay, good. All right. So now it's time to put it all together. Okay, I'm going to give your brain a little bit of a workout, perhaps. Okay, so how do T cells get activated? Remember, when those cytotoxic T cells get activated, they are going to act to kill one of our cells that's infected with a virus. So this is not from your book. It's a little bit simpler than the diagram from your book. So let's look at this first. Okay, so here's a macrophage that is ingesting a virus. More correctly, a virion, right? Because it's outside the cell, it's got its capsid on it and everything. Okay, so it's ingesting a virion, and it's chopping it up in the phagolysosome, and it's going to be presenting that antigen by MHC class 2. And hopefully, again, this is happening, most likely it's migrating through the lymph, maybe in a lymph node, and it's going to hopefully come in contact eventually with a helper T cell, with a T cell receptor that has specificity for that same antigen. Okay, so that is going to activate that helper T cell. Okay, but remember, it's the cytotoxic T cell that actually is going to do the work. When a cytotoxic T cell comes in contact with the same antigen presented by an infected cell with MHC class 1, okay, that synapse is not enough, even though you think, oh, it might have taken a long time for that T cell receptor to find that antigen presented by MHC class 1. It needs signals from that helper T cell, cytokine signals. Don't worry about that it's IL-2. Cytokine signals acting on it now to make that cytotoxic C cell proliferate into more with exactly the same specificity. And now it's ready to act on those infected cells and destroy them. Okay, and we'll talk about how it does that. Okay, your book's graphic just adds a couple of details that I want to point out. Okay, so here it starts with a dendritic cell. Okay, and of course, a dendritic cell is phagocytizing this antigen maybe and presenting it with MHC class 2 to that T helper cell. That T helper cell, when it activates, it actually differentiates into a T helper 1 cell and to release the right cytokines. At the same time, an inactive cytotoxic T cell, here's a blow up of the immunological synapse that we see there. So the cytotoxic T cell with the T cell receptor that recognizes the epitope presented by the dendritic cell. Don't forget about that CD8 glycoprotein that cytotoxic T cells have. That actually binds to the MHC glycoprotein to kind of stabilize that synapse. But remember, that's not enough. That cytotoxic T cell gets these cytokines from the helper cell that also recognize that to say, yes, this is an appropriate action to take. You want to make sure that those cytotoxic T cells aren't just killing cells willy-nilly. These are our own cells, right? So when it gets the right signals, it's going to undergo clonal expansion. That means it makes many more of itself, divides many times, and you also get some memory T cells. So that's important because when that maybe years later, when that memory T cell recognizes the same antigen, it's going to activate right away. And it's not going to need those signals from the T helper cell. Okay, that takes time for all those signals to be generated. And it's going to activate those cytotoxic T cells that were just made with that receptor. I think it's kind of funny the way they drew this. It always looks like the cells are smoking a cigarette or something, but that just means they've got receptors for the cytokines that it self-stimulates. So basically, the cells make cytokines to stimulate itself to act on those virally infected cells. Okay, so these are active now. What are they actually doing? We could go through and yeah, let's go ahead and label this. Okay, here's the cell that's infected. And here's the cytotoxic T cell. Okay, here's the CD8 T cell receptor. Here's the epitope. And this is the antigen being presented by MHC class 1. Okay, so now we have a active cytotoxic T cell. Okay, so once it's active, okay, how is it going to work? The book talks about 2 different ways that the cytotoxic T cell can bring about death of the pathogen. We're not going to worry about one. We think this one's maybe a little bit more common, the perforin granzyme pathway. So the other one, that CD95 pathway, you don't need to worry about that. These are 2 cytokines that are released by the activated cytotoxic T cell. Okay, again, 2 cytokines. Perforin does what it sounds like it would do. It perforates that host cell. So it pokes holes in the membranes. Remember, our cells don't have a tough cell wall around it, so this is fairly easy to do. Pokes holes in membranes. And that allows the entrance of the second cytosine, granzyme, and granzyme induces apoptosis or apoptosis, however you say that. Okay, that's a programmed cell death. So basically, a lot of things we don't understand about that mechanism, but granzyme just induces our host cell that's infected with a virus to commit suicide, basically. Okay, we don't really want that cell to lyse because we don't want to release all these virus particles that might be reproducing in it. It just kind of, it's a programmed cell death. Okay, so once one of our cells is infected with the virus, there's really not much that can be done except sacrifice that cell and hope that more other cells or aren't infected. Okay, questions? Okay, good. Good, all right, so we're getting there. So how do B cells get activated? Remember, when they're activated, they're gonna make antibodies, differentiate into plasma cells and make antibodies. We said that there's two ways that that can happen. In one way, the cell can be activated by the antigen itself. So it doesn't need extra stimulation from T helper cells. So we call that, we call these antigens T cell independent antigens. These are antigens that can activate a B cell to make antibodies by themselves. Okay, so this is a little bit simpler, right? What kind of antigens are able to act this way? They're mostly large antigens that are not complicated. They have lots of repeats. So some kind of polysaccharide, where it's, again, the same monosaccharide over and over again. And we said earlier that these kind of molecules generally are not very good antigens, but there are exceptions. All right, so oftentimes we see these kind of polysaccharides in bacterial capsules. Okay, so the nice thing about B cells getting activated this way is that it's really fast. You don't have to wait around for all these, you know, this T cell receptor to find this and that, and you know, we don't. It's fast, right? You get antibody pretty quickly, but there are a couple of problems with it. We get a little weaker response because only Igm antibody is produced. Okay, so in other words, there's no class switching to some isotype of antibody that may be able to better conquer the pathogen. You only get Igm. And there are no memory cells produced. So if this is a really fast response, maybe that's not a problem, because again, the idea with memory cells is activation can happen right away. But there's no memory. And then the third problem is that young children don't have a very good response to these antigens. So kids maybe up to age 2 or 4, perhaps, because their B cells are very immature, they don't recognize these kind of antigens very well. Okay, so T cell independent antigens can, act. Young children are a lot more susceptible to them. So they cause infections in young children that as adults we wouldn't get. So we're going to mention a couple of these later on, and we're talking about specific pathogens, but one of them is Haemophilus Influenzae. The capsule of that bacteria is a T cell independent antigen, and that can cause a bacterial meningitis in young children. So it's a very serious infection, can cause, disability, death even. This actually, its name, it doesn't cause influenza. 100 years ago in that pandemic, we didn't know what was causing flu back then. We thought it might be this organism. And that's why we called this, gave it this species influenza. Luckily, we have a vaccine to this. If you've ever heard of the Hib Vaccine, it stands for Haemophilus Influenza type B. So it's really important that babies get this series of Hib vaccines. You might say, adults might think, well, we never get that. They don't need that. But, again, babies can get it. So it's important to vaccinate against that. Okay. All right. So much more common. Oh, let's illustrate that first. Okay. So again, B cell receptors come in contact with these polysaccharides. Again, some bacterial capsules can, that's a good example of what can act as T independent antigens. And then the B cell will differentiate into plasma cells, and kick out IgM antibodies. For some reason, they didn't draw those as a pentamer like IgM antibodies usually are. But then remember, no memory cells over here. Okay, so it's a little bit of a weaker response, but again, it's fast. Okay, we think much more commonly the antigens that we come in contact with, we need some stimulation from the T helper cells in order to stimulate our B cells. Okay, so we call these T-dependent antigens. Okay, so the way this works, say we start with some antigen presenting cell like a dendritic cell, and it is has phagocytized some antigen and presenting bits of that with MHC class 2. Okay, and hopefully carrying that to the lymph node to hopefully come in contact with the T helper cell or the T cell receptor that recognizes that epitope. Okay, so we know this is a T helper cell because it has the CD4. Don't worry about these other receptors. We're not going to worry about what they do. That's really small. Blow that up a little bit. Okay, so once that synapse has been made, now this T helper cell is going to be activated or to differentiate into T helper type 2 cell. Okay, so remember it was T helper type 1 to act on the cytotoxic T cells. Okay, at the same time, we've got a repertoire of B cells with all different B cell receptors. When a B cell receptor recognizes that same epitope, B cells have a way to kind of ingest antigen. We don't really say phagocytize, maybe there's some difference perhaps, but it will ingest and present that same antigen with MHC class 2. Okay, and then those B cells, I'm looking over here at this synapse, will hopefully come in contact with a T helper 2 cell. And when that synapse is made, then the correct cytokine signals are generated from that T helper cell to cause the B cell to differentiate into plasma cells with that same specificity, as well as some memory cells. And again, they'll start kicking out especially the IgM antibodies. Okay. Also within your homework is an animation to walk you through the basics of that again. Whew, okay, so the so what of that, all right, we'll finish this, we're almost done here. So this is going to lead into our discussion of vaccines on Monday. But the idea is that say a patient that there is a difference between the first time a patient will be exposed to an antigen and subsequent times. So if we have, assuming this is a T-dependent response, or we'll say T-dependent antigen. Okay, so like a tetanus shot. Okay, a tetanus vaccine. Basically a tetanus vaccine is just the toxin from tetanus that has been deactivated. So say a patient gets a tetanus shot. Okay, what happens in their bodies? Well, around day maybe 6 or 7, it takes a while for all those, T helper cells to find the right B cells. It'll start making IgM. By class switching, eventually maybe a little bit higher titer of IgG. That even takes time, takes a couple weeks. Usually they say when you get a vaccination, it takes a couple weeks till you're protected. Okay, all those immunological synapses take time. But the idea is that at some later date, if that same patient is exposed to the real thing, tetanus toxin, now because that patient has memory cells to that antigen, then it starts kicking out antibodies much sooner, maybe half the time, and different kinds of antibodies at the same time in much higher titers. Okay, so more antibody, the right kind of antibody, and quicker the second time it's exposed. And maybe the third time it's even a little bit faster and a little more. Okay, so that's the advantage of memory cells. They require less stimulation to activate both memory B cells and T cells. I didn't mention this before, but in as well as isotype switching over the course of infection, we do get a little bit of affinity maturation, meaning those antibodies, those B cell receptors will undergo maybe some point mutations and eventually kind of learn to fit that antigen a little bit better. Okay, so that we get kind of a little better response as time goes on. Okay. All right. So this is basically a similar diagram from another source. Just drew it out a little bit different. You can't really tell the timing very well here, but primary and secondary responses against T-dependent antigens. Think about how would this be different if the antigen was not T-dependent, if it was a T-independent antigen. If this was our first response, what would our second response look like? How would that be different? Yeah.
There wouldn't be memory cells, so it wouldn't like do it faster.
Right. Good. So because there's no memory cells, it wouldn't make antibody any faster, and it wouldn't make more. Okay. So this is the slide that I didn't post. Right. If it was a T-independent antigen, we would get the same response the second time. It's kind of hard to tell the timing from the way they put it there, but you know, same kind of antibody, same amounts, the same time. Okay, because there's no memory. All right, good. I think I filled your brains up enough. I just have one little thing to finish up with there, and that's going to again dovetail really nicely into our discussion of vaccines. So I'll get that material posted. Any questions? Yeah, question. Okay. Oh, this one, wait. That one? Oh, I know. It looks like I meant to write something, but I think I just wanted to kind of underline, okay, now we have an activated cytotoxic T cell. So you could make a note of that, hey, now it's activated, it's made this connection, and then it's going to start kicking out those cytokines. Yeah. Okay, good. Have a good day. Hope you stay dry.